everything should be there. And just know that tomorrow you do have a final review session with your SI leader, Vasily. That's at 6 p.m. in the student union as well. I think it's in Egmont Key. So if you're looking for a little extra help and a little extra opportunity to review, please go see Vasily tomorrow. I'm sure he'd love to have you and he'd love to help. If you're just walking in, there is a stack of evaluation sheets over here on this table. If you would like to grab one and fill it out just to, talk, give us a little evaluation about your experience, please feel free. Those are over there. And again, if you don't already have your review packet, you can find it. Just follow that, and that will give you the link to where you can download that review packet. We'll just be filling it out. So this session is going to be a little different than normal. I'm going to go at a pretty fast pace because I already have them all filled out for you. If we're going to get through all 27 pages, I'm pretty much just going to be going through it. So what I'd like to say is if you do have questions, please make a note of them, and I'll try to finish so that we have time to address those questions at the end. But just to get you guys through this material, I kind of have to go at a breakneck pace, unfortunately, so you only have 2 hours. If you don't want to walk back to your car in the dark alone, there are 2 numbers here, and these will be escort services for you. They'll bring you a security officer or literally drive a golf cart to you and take you to your car if you would prefer that. So if you would like to take down these numbers, go ahead and take a picture or write it down. And other than that, we can get started. Are we good? Yeah, all right, let's start. So, unfortunately, this is the first time I'm using a .cam, so I apologize if this is a little rough, but I'm going to try and make the most out of this as I can. Let's get this to focus. Does it look okay for everyone? Is anybody having a hard time reading that? I'll read them out loud anyway. But let's go ahead and get started. I'm going to actually get a little closer. There we go. It's a little better. Oh, zoom in. Wow, okay. So we're going to start with chapter 15. And we'll start at the top. So, endocrine system, nervous system, they're the major information centers between different cells and tissues in our body. And so what Dr. Angari likes to say is that our hypothalamus is the president of our endocrine system. It's the main control center. And it has connections to the pituitary gland at the inferior hypothalamus. And we'll talk a little bit more about how those 2 areas are connected and how they start sending chemical signals to each other. If we can get this to focus, please. Oh, wow, great. This .cam is a little finicky, I apologize. Let's see if we can get this back up. So I'm just going to keep reading then. So hypothalamus, president of the endocrine system, inferior region contains the pituitary gland. The pituitary gland itself is what is considered our vice president of the endocrine system, and it has 3 lobes, our anterior, intermediate, and posterior. And then your quote-unquote citizens are going to be your peripheral glands, and those are the glands that will take these hormone stimuli and figure out what to do with them. Either they will react in a way, or they will actually be stimulated to secrete their own hormones. And these citizen peripheral glands include the adrenal gland, thyroid gland, our ovaries, and our testes. We can get back into the mechanisms of the hormone secretion. So we have endocrine hormones, a chemical substance which is produced and secreted by the cells of the endocrine gland and released into the bloodstream, which carries it to its target cells. Endocrine hormones all require specific receptors on their target cells. Without these receptors, they can't bind to the target cell at all, so they will have no effect. And the cell will not carry out their physiological reaction to the hormone signal. Whether or not that's stimulation or inhibition really depends on the receptor, the target cell, and the hormone. Exocrine hormones are chemical substances produced in the exocrine cells that are not circulated into the blood. So these hormone signals are going to be secreted into interstitial fluid, to neighboring cells. They can be secreted onto a tissue surface or into a cavity. So examples of this would include pancreatic enzymes, which are secreted by our pancreas, of course. We've got saliva secreted by our salivary glands, which include our sublingual, parotid, and submandibular glands. Those are important. And of course our sweat, which is an example of something being secreted onto a tissue surface, is secreted by our sweat glands. Now, exocrine hormones are produced in the acinas, and these are little structures produced by a collection of acinar cells, A-C-I-N-A-R, acinar cells, to produce an acinus, and they're secreted out of the gland by ducts. So exocrine glands have ducts. That's how they excrete their hormones. So pancreatic ducts, common bile duct, parotid duct are all examples of this. And these ducts will open onto the cavity or surface of their target organ and tissues. Now we have some actual examples of different types of exocrine hormones. The first one being paracrine hormones, which stimulate or inhibit the actions of neighboring target cells. So any cells that are nearby, those target cells may or may not produce hormones themselves as a result of that. And a good example of that would be somatostatin, being produced in what we'll find are delta cells to inhibit insulin secretion in pancreatic beta cells. So we'll see that a little later on. Autocrine hormones are going to be giving feedback to their self. So they'll be secreted outside of the cell, and then they will return to their own cell surface and bind to cell receptors there. So they'll be giving feedback to their own cell to stimulate or inhibit its cellular activity of their own. And a difference here between autocrine and intracrine, they both stimulate themselves, but intracrine hormones do not leave the cell. They do not pass the cell membrane. So intracrine hormones will stimulate or inhibit cellular activity inside their own cell without being secreted. So they don't need to leave and then rebind to their own surface. A good example of this would be insulin. So they can actually, let me put that back up there. They can actually regulate themselves inside their own beta cells in the pancreas. And that's pretty much it for exocrine hormones. We can move on a little bit to hormone synthesis. So protein hormones are going to be produced the exact same way as any other protein is going to be produced in our cells. There's no real special mechanism or machinery in our cells that protein hormones need. They are produced the exact same way as other proteins. Peptide hormones, however, even though they do derive from larger proteins, they do undergo an extra step. So peptide hormones are going to be activated by being cleaved off of a larger protein. So oftentimes those proteins will already be formed, and so to activate that peptide hormone, they have to be cleaved from that larger protein molecule. So produced from cleaving off a portion of larger protein precursors. This includes insulin, ACTH, and glucagon. Last one, well no, second to last one, thyroid hormones are produced by organification and coupling. Organification is going to be the process where we combine iodine and tyrosine residues. Those tyrosine residues are going to come from thyroglobulin proteins in the thyroid gland. We'll see that later on as well. And then coupling is going to follow organification, which is just the different molecules that are produced by organification being combined. Those different combinations will give us either T3 or T4. We'll go over that mechanism as well. Steroid hormones, last one, produced from cholesterol precursor molecules. So when we're talking about protein hormones versus steroid hormones, there are a couple different properties that you're going to need to know. One of those is binding sites. So protein hormones are going to be hormones that bind to cell surface membrane receptors, while steroid hormones and thyroid hormones, you can make an addendum to that, steroid and thyroid hormones are going to bind to nuclear or intracellular receptors. So they're going to pass the cell membrane, go to the target cell nucleus, and then have their action there. Next page. So, some more properties. Protein hormones, if we're talking about, let's get them there. Can you see that? A little better. Protein hormones are going to be lipophobic, which means that they are lipid repulsed. And then steroid hormones are going to be lipophilic or lipid loving. That's not very clear. And then if we're talking about the way that they react to water molecules, polar molecule, protein hormones, being polar, are going to be hydrophilic or water-loving. That is really not very, that might be a little better right there. Can you see a little better? Yeah. And then steroids are going to be hydrophobic or water-repulsed. If we're talking about transport in the blood, since these are protein hormones, steroid hormones, and thyroid hormones are all, gosh, that looks terrible. Endocrine hormones, they'll be carried in the blood. Protein hormones specifically do not require a carrier and can circulate freely in the blood. This is because they're proteins. So steroid hormones and thyroid hormones, therefore, are going to require specific carriers to carry them through the blood in order for them to reach their target cell. And these carriers, these transport molecules, are usually proteins. Now, the duration of the hormone action on their target cell, the length of time it takes for that hormone to bind and activate at that receptor. Protein hormones are going to have a short action time because, again, they have no transport or carrier molecule. Therefore, steroid hormones and thyroid hormones are going to have a longer action period because they first need to dissociate from their transporter or carrier molecule before then binding to the receptor. So, if we're talking about molecular precursors to these hormones, protein hormones, their precursor is an amino acid sequence. So amino acids are going to be the building blocks for proteins and also peptides. So any amino acid that is specific for those hormones, those are going to be the precursors. Steroid hormones, on the other hand, we already said, cholesterol precursor. Now thyroid hormones is where we get a little bit more specific. They have an iodide ion precursor. And I know I had in parentheses an amide. I just crossed that out because that is not applicable here. That is a mistake. The amino acid, what I meant to put there was that it's an amino acid tyrosine residue. Tyrosine residues are also considered a precursor for thyroid hormones, along with the iodide ion. So now we can talk a little bit more about receptors. We have cell surface receptors. Those obviously are going to be located on the target cell's membrane surface. So those receptors are embedded in that surface membrane. And those will be specific for protein and peptide hormones. Protein and peptide hormones like to bind to cell surface receptors. And a good example of that is insulin, growth hormone, or prolactin. And these examples are including but not limited to these hormones. This review guide, I know it's very long at 27 pages, but it's still not 100% complete. So I would say add your own kind of stuff to it if you would like. But keep in mind that not everything here is just 100% the amount of information. So just supply or add the little parts that you find are missing from your own notes. But I did try to be thorough. Nuclear receptors, on the other hand, are intracellular receptors, intra meaning inside the cell. Those are located inside the cell at the nucleus specifically. And those are going to bind steroid hormones and thyroid hormones. An example of that, testosterone, estrogen, androgen, progesterone, aldosterone, cortisol, T3, T4, and vitamin D. So that's a lot. Now we can go into effects. When two or more hormones have similar functions and effects on their target cell, we say they are synergistic. They work in harmony for a specific outcome. An example of that would be aldosterone and norepinephrine, both increasing blood pressure, or cortisol and insulin, both regulating blood glucose levels. They work for the same outcome. A permissive effect would be simply when one hormone gives quote-unquote permission for the action of a second hormone. This can mean that it enhances the responsiveness of the target cell receptor to that hormone, or it can increase the activity or reaction of that target cell to the hormone binding. So that could be an increase in secretions versus a normal amount of secretions. And then we can go to antagonistic effects. We've got to zoom. Here we go. Antagonistic effects, when the binding of one hormone actively inhibits the effects of the other hormone. This is typically used when designing drugs. We see a lot of synthetic hormones that are designed as antagonists for a specific receptor if those receptors or that hormone production is excessive. And those antagonists will actively bind to the same receptor and then block it so that that hormone can't act there. So it inhibits a process that may become pathological. For example, propranolol is an antagonist for our beta-1 receptors by binding and blocking norepinephrine from being able to bind at that receptor. So it'll bind to beta-1, and because it binds to beta-1, norepinephrine cannot bind. That's an antagonist. Agonists, on the other hand, are synthetics or sometimes even other hormones that will bind to a receptor to act as that original natural hormone. So we say they act like natural hormones and bind to natural hormone receptors. An example of this would be hormone replacement therapy. If somebody is deficient in growth hormone, those receptors can be bound by synthetic hormones in order to activate those target cells. So an example of that happening would be dwarfism, where there's a deficiency in growth hormone. We can treat that with synthetic growth hormone to stimulate that receptor. So now that we've talked a little bit about receptors, let's get into some detail about the specific 4 types of cell surface receptors you're going to see. So our first one is our G protein coupled receptor, GPCR right here. G protein is an intracellular protein with connected to a receptor protein with a 7 transmembrane domain. What that means is that there is a receptor, and then the G protein itself is inside the cell. But that G protein itself, the way that it's folded up, that sequence, that long protein molecule, folds and goes through, in and out, through the lipid bilayer of that cell membrane 7 times. So it's kind of coiled up. The receptor, the protein receptor, its amino acid, or not amino acid, I'm sorry, our amino terminal, or N-terminus, is exposed to the outside or extracellularly, whereas the carboxyl, or C-terminus, is going to be exposed intracellularly, and that's just saying which pole of the protein is inside, which pole of the protein is outside. Besides that, our G protein has 3 subunits attached to it inside the cell. And those 3 subunits are going to be our alpha, beta, and gamma. I know we've seen specifically the activation of these alpha subunits when we were talking about the sympathetic nervous system, if you remember. So when a hormone binds to this receptor, that G protein alpha subunit specifically is going to be able to bind GTP, or guanosine triphosphate. GTP acts like ATP and provides energy by cleaving off that 3rd phosphate. So intracellularly, that alpha subunit is going to use its own GTPase activity in order to cleave that 3rd phosphate and use that energy to become activated. In order to inactivate itself, that alpha subunit can also bind GDP, or guanosine diphosphate. Without that triphosphate there, it won't be active. So it can actively bind GDP in order to shut itself off. So G proteins, those alpha subunits can either be stimulatory or inhibitory, again like we saw with norepinephrine in the sympathetic nervous system. G proteins can be coupled with other effector molecules that pass on that activation signal. to what we call a second messenger. So examples of effector molecules would include adenylate cyclase and phospholipase C. These molecules then move on further into the cell to activate second messenger molecules. And good examples of second messenger molecules would include IP3 or inositol 1,4,5 triphosphate, inositol 1,5,4 triphosphate, IP3. Another example would be CAMP, CGMP, calcium, which then binds and activates to a molecule called calmodulin. But calcium itself is the second messenger. Together we just call it calcium calmodulin. So, some examples, but not all examples, of molecules that bind to GPCRs. We've got catecholamines, which includes our noradrenaline and adrenaline, meaning our norepinephrine and norepinephrine. Prostaglandins, ACTH, glucagon, PTH, and TSH. Those are some examples. And I have this example for you in the notes, but since this is filled out and doesn't have any blanks, I'm just going to go past it. You can read it on your own, but this is just a review of that sympathetic nervous system action at that alpha-1 receptor and subunit. So that's just a review of that information from your nervous system block. If we move on, we can talk a little bit more about the second type of cell surface receptor we have, which are growth factor receptors. Do not mistake growth factor for growth hormone. Those are two different things. We'll talk about growth hormone in a second. Growth factor receptors, this is another protein receptor, and the amino terminal of this protein is going to be exposed outside of the cell, or extracellularly again. And that extracellularly exposed amino terminal can act as a binding site. This growth factor receptor protein only has one transmembrane domain, which means that that protein as a whole only passes through the lipid bilayer of the cell once. That's it, just once. Now its C-terminus, or carboxy terminal, is going to be on the intracellular side of the cell, or inside the cell, and that C-terminal will actually act as a catalytic domain. That means it has some sort of enzymatic activity. And what we see here as that activity is autophosphorylation. So it picks up, this camera, it picks up phosphate and it will activate itself that way. It will kind of generate its own energy. So it'll phosphorylate itself in a process called autophosphorylation. And then it will activate second messenger tyrosine kinase by passing on those phosphates to the tyrosine kinase. This occurs when the hormone is bound. So examples of that are receptors, growth factor receptors will bind insulin, insulin growth factor, and epidermal growth factor. And keep in mind that IGF and EGF are hormone cytokines involved in cell proliferation and cell growth. So that means that the cells will increase in number, and then an individual cell can also increase in size. Next cell surface receptor is a cytokine receptor. So these are specific for growth hormones. So again, growth factor, growth hormone, not the same thing. Growth hormone does not bind at growth factor receptors. They will bind at cytokine receptors. So cytokine receptor proteins have their amino terminal exposed extracellularly outside the cell to act as a binding site. And again, we see that this protein only has one transmembrane domain, meaning it passes through the lipid bilayer only once. That carboxy terminal end of the protein, if I can get this to focus, is exposed intracellularly. I guess that's as good as we're going to get. Inside the cell for activating its second messengers. When growth hormone binds to the receptor, that association between that activated receptor's carboxy terminal on the inside and its second messenger, JAK2, so this molecule is already associated and will activate once something is bound. That JAK2 effector molecule will then have a conformational change in its protein structure, which means its shape will change. And that will activate its own enzymatic activity, which we call tyrosine kinase catalytic activity, which pretty much tells us what it's going to do. It's going to activate tyrosine kinase, which begins then the signal transduction through the rest of the cell until we get a reaction. So, fourth and final cell surface type of receptor is our ligand-regulated transporter, or a guanylyl cyclase receptor. So these proteins are proteins that change shape when something is bound. These receptors will change shape, and the ligand is the hormone in this case. Ligand just means something that can bind. And then this will usually be a channel for ions, and since ions will be flowing through that channel and into the cell, we regard those ions as second messengers. So ions that flow through will be considered second messengers. Nitric oxide is a good example of this. Nitric oxide can act as a hormone, it can act as a neurotransmitter, it can do a bunch of different things, but right here we say that nitric oxide is a vasodilator, causing an increase in intracellular nitric oxide synthase enzyme, which will then stimulate guanylyl cyclase, GC activity, which is similar to adenylate cyclase. So where adenylate cyclase gives us CAMP for intracellular energy, guanylyl cyclase will give us CGMP for intracellular energy. And this will then activate protein kinases for vasodilation in the penile dorsal vein during erections. That's something we can get into a little bit later. And then there's another example here, ANP, atrial natriuretic peptide. I have that example already filled out for you. So we can move on to nuclear receptors. So these receptors are very specific for steroid hormones and thyroid hormones. We can also have vitamin D using these types of receptors, retinoids, fatty acids, and bile acids. So these receptors are also proteins. They're just kind of stuck through the nuclear envelope around the nucleus. So those receptor proteins have an amino terminal that will have a high amino acid variation, which means they're very different. And the reason that they're so different is because they have to find specific genes in your DNA that hormone binding will target for transcription and translation. So the receptor will then also have a DNA binding domain as part of its protein structure so that it can find the DNA that it wants to transcript and transcribe and translate. And then a carboxy terminal of that receptor protein will act as a binding site, as well as being able to control the transcription of a target gene. So, again, these receptors control gene expression. They can bind to DNA on the promoters of target genes or to the transcription factors of target genes. So, they can directly control the transcription of a target gene, or they can upregulate something that's already being transcribed. So they can increase that transcription. So binding of a signal can stimulate an increase or decrease in target gene transcription translation into a protein product meant to be used for some function. And that can be used inside the cell, or it can be ultimately exocytosed to fulfill its purpose, whatever that might be. Now, if we talk about the way that steroid and thyroid hormones actually work, we can say that a steroid or thyroid hormone would first diffuse across the target cell membrane. That hormone would then bind to the cytoplasmic site on the nuclear receptor protein. So it's intracellular, but it's inside the cytoplasm of the cell, not inside the nucleus. Then the nuclear receptor will undergo shape changes, conformational changes, to expose that DNA binding site of the target gene. That target gene will then be transcribed, resulting in messenger RNA, mRNA. That sequence of the target gene will then be translated to produce a specific protein product that will satisfy the cellular physiological reaction to the binding of the initial hormone signal. So we're going to get a product out of all of this. Hormone will bind, and we will get a product in return. And the purpose of that product could be anything, depending on what we're talking about specifically. So let's talk a little bit about endocrine secretions and their functions. The hypothalamus itself is an endocrine organ that controls a lot of things. You talked about it a little bit in your immuno chapter with fever, temperature regulation. It controls our sleep, our memory, learning, emotions, and sexual behavior. And it also has connections and control with the autonomic nervous system, and it itself pretty much makes up our endocrine system. It's the president, like we said. So, first hormone we can talk about, this corticotropin-releasing hormone, CRH. The blood vessel connecting the hypothalamus and our anterior pituitary gland is this HPPS, hypothalamic pituitary portal system. Hypothalamic pituitary portal system. And this HPPS will allow for the movement of the CRH from the hypothalamus and into the anterior pituitary. And if we see this RH right here, releasing hormone, our hypothalamus will be giving that pituitary releasing hormones. So releasing hormones come from the hypothalamus, and they go into the anterior pituitary, which contains very specific trophic cells. So usually these hormones will be going towards a specific trophic cell, which are usually named after the hormone that's being secreted, to stimulate them to then secrete stimulating hormones. So CRH will be released from the hypothalamus into the anterior pituitary, and it will bind onto anterior pituitary corticotropic cells. Corticotropic cells. When CRH binds to these cells on their membrane surface, they will activate CAMP as a second messenger inside the cell, and that cell will then secrete adrenocorticotropic hormone, ACTH, adrenocorticotropic hormone. ACTH will then be carried by the blood out of the anterior pituitary and to the adrenal cortex of the adrenal gland in order to bind on a adrenal cortex cell surface G protein coupled receptor, GPCR, with CAMP as a second messenger to stimulate the secretion of androgen, aldosterone, and cortisol. We have a little bit on those three here. Androgen is a sex hormone seen in males and females in smaller amounts, and it's important for the beginning of puberty in females. This specific hormone, androgen, is produced in the zona reticulata layer of the adrenal cortex. They bind to nuclear receptors, and it is involved in axillary armpit or pubic hair growth and also acne. So if you have an excess in acne, you might have an excess in androgen. Aldosterone helps us to increase the salt concentration and water reabsorption from our nephrons in order to increase our own blood pressure and our blood volume. Aldosterone is also called a mineralocorticoid, and it is produced in the zona glomerulosa of the adrenal cortex. It will bind to a nuclear receptor in the cells of the collecting tubule of the nephron. It is involved in our renin-angiotensin system, and again, it increases absorption of salt water. It also increases bicarbonate absorption to increase our blood pressure, and increases the secretion of hydrogen ions or acidic molecules and potassium into the urine. And it was usually activated when blood pressure is low. We do have the mechanism for that as well later on. Cortisol is a stress hormone. It's also called a glucocorticoid. Maintains blood glucose, metabolism, and appetite, and is produced in the zona fasciculata of the adrenal cortex, zona fasciculata. It's got a nuclear receptor, helps us to degrade proteins, which is proteolysis, suppresses our immune system and inflammation, which is why if you are very stressed out, you are more likely to get sick. It also increases lipolysis, which is our lipid degradation, and stimulates the formation of new glucose, gluconeogenesis. Second hormone we can talk about, TRH. Again, we see that RH right there, releasing hormone coming through the hypothalamus and into the anterior pituitary to stimulate thyrotropic cells. So thyroid or thyrotropin releasing hormone, thyrotropic cells is what they stimulate. And those cells will be stimulated to secrete TSH, our thyroid stimulating hormone, which then enter the blood to stimulate thyroid gland to secrete T3 and T4. And it also stimulates those cells in the anterior pituitary to secrete prolactin as well. So TRH, thyrotropin-releasing hormone, results in TSH and prolactin. Now, prolactin in females helps in the development of mammary glands for breastfeeding, but in excess can cause infertility because it will actually suppress other sex hormones. But prolactin is important during pregnancy because it can inhibit the menstrual cycle from taking place while you're pregnant, so you don't have a miscarriage. In males, it helps control emotional and sexual behavior. Can cause infertility as well if it's in excess because, again, it suppresses sex hormones. Other things we can talk about from prolactin is that dopamine actually inhibits it. Dopamine, which also comes from the hypothalamus. And prolactin uses JAK2 as a secondary messenger. JAK2 as a secondary messenger. So TSH will bind to follicular cells in the thyroid gland. This is a GPCR, or G protein-coupled receptor, that it binds to on the cell surface. And it uses CAMP as a secondary messenger. Helps control fetal thyroid gland development, and helps with the development of the central nervous system, the brain, and the bones. So T3, T4. TSH, thyroid-stimulating hormone, will stimulate the production of T3, T4 via the iodide pump. So iodine will result from that. We get iodide ion symporting in with sodium. We'll talk about that later. And we result with iodine, I2, and tyrosine residues from thyroglobulin proteins already inside the thyroid gland will then combine during organification and catalysis by peroxidase. Peroxidase enzyme will catalyze this to form MIT and DIT, which we can then combine in the coupling reaction to build T3, T4. T3, T4 use nuclear receptors and have roles in memory, learning, metabolism, heart rate, contractility, blood pressure, and temperature, and a lot of other good stuff that's listed there for you. So GnRH is our gonadotropin releasing hormone. Again, we see that RH releasing hormone from the hypothalamus. And that GnRH will stimulate gonadotropic cells in the anterior pituitary to secrete follicle-stimulating hormone, FSH, and LH, luteinizing hormone. So GnRH uses inositol 1,4,5-triphosphate, or IP3, as its second messenger. FSH and LH in females. FSH levels will increase during the first 2 weeks of the menstrual cycle, and this is what we call the follicular phase, in order to mature and develop follicles in the ovaries into corpus luteum around a single egg or ovum. Once they enter the mid-cycle, or mid-13, 14, 15 days, right before the second phase, FSH level will drop and the LH level will increase because the LH luteinizing hormone will then begin ovulation as the last step of follicle development, where that egg will actually rupture the follicle and burst out of it to then leave the ovary. So luteinizing hormone is important in that egg leaving the ovary via rupturing through the follicle. So FSH and LH in males. FSH will have some cell receptors in the testes to control spermatogenesis, which is our germ cell to sperm maturation process. So spermatogenesis followed by spermiogenesis will be under the control of FSH. But when FSH is overactive and we have sperm production that's too high, Sertoli cells in those testes will then secrete inhibin to prevent FSH from sending its own feedback to the anterior pituitary. It has a positive feedback mechanism that it can tell its own cells to secrete more of itself. back in the anterior pituitary. So inhibin can actually stop it from doing that. And in that way, we have regulated spermatogenesis. Luteinizing hormones will have receptors on more specific cells, Leydig cells, Leydig. So LL, luteinizing, Leydig, those two are together. LH binding to these receptors will stimulate the secretion of testosterone, which is then used in reproductive regulation and puberty, so the formation of our secondary sex characteristics. And the information we have for surface receptors, FSH will bind to GPCRs, G protein-coupled receptors, and use CAMP as a secondary messenger. Now we have growth hormone, releasing hormone, GHRH, secreted by the hypothalamus into the anterior pituitary to bind to the G protein-coupled receptors on the surface of somatotropic cells. This is the only one where its trophic cell is different from the hormone name. So growth hormone, because growth hormones are general and will affect our body growth as a whole, will then affect somatotropic cells, because somato means body, soma, so the growth of our body. And that will stimulate secretion of growth hormone, GH. GH will then also be secreted by the placenta in pregnant females, and it will use IP3 and CAMP as secondary messengers inside the cell. So growth hormone will control cell proliferation, so an increase in the number of cells, and cell growth, or the control of cell size, in the ovum upon fertilization. So that is what's controlling the creation of a blastocyst or our embryo from a single-celled zygote. GH will bind to cytokine receptors and use JAK2 and tyrosine kinases as secondary messengers. Again, growth hormone is not growth factor. So growth hormone has cytokine receptors. We can talk a little bit about the hypothalamus. Now, our hypothalamus is connected to the pituitary gland in two very distinct ways. And it can be connected to the anterior or posterior pituitary gland, depending on which part of the pituitary kind of changes what kind of connection we're talking about. So if we're talking about the hypothalamus and the anterior pituitary gland, blood vessels will actually facilitate the movement of hormones secreted in the hypothalamus in order to send them to the pituitary. And that's what we call our hypothalamic pituitary portal system, HPPS. And those are tiny little sinusoid vessels. We also have the primary capillary plexus that connects the hypothalamus and anterior pituitary. So that's the anterior lobe. Releasing hormones are specific for this area. They'll be released from the hypothalamus and sent to the anterior pituitary to then stimulate trophic cells. Those trophic cells will then release specific stimulating hormones like we saw TSH. Not every single one will have that SH at the end, because we saw ACTH. That doesn't have it. Prolactin doesn't have it, but those are still considered stimulating hormones. And then those will leave the anterior pituitary and do their thing in the peripheral tissues. Second way that we can have the hypothalamus connection to a pituitary gland is to the posterior pituitary gland, and that is more nervous system control between those two areas. So we have the supraoptic, and paraventricular nuclei, and those nuclei bodies will be found in the hypothalamus, and they will have fibers extending down into the posterior lobe of the pituitary gland. Once they're there, those nuclei fibers can be stimulated, and those axon terminals will be secreting oxytocin and ADH in the posterior pituitary. They can secrete it fresh, or they can secrete it there to store it there, either way. And then oxytocin and ADH will be secreted out of the posterior pituitary and into the blood system to do its thing at the peripheral tissues. Now, I mentioned a little bit about our positive and negative feedback systems. So when we see peripheral hormone levels decrease, those peripheral hormone decreases can send a positive feedback to the central hormones, SH and RH, in the hypothalamus and anterior pituitary to increase their secretion. So when peripheral hormones increase, it can send a negative feedback to central hormones in order to decrease their secretion. So if they're excessive, they can send a stop signal. If they're too low, they can send a go signal for secretion. So talk a little bit about the pituitary gland. We have 3 lobes, anterior, intermediate, and posterior. Really, it's just anterior and posterior, but there is an intermediate kind of like chasm where those tissues kind of are together, and they do very specific things. So they do have trophic cells that are stimulated by the hypothalamus, as we said. They secrete specific hormones, bind hormones. Here I have examples of anterior pituitary hormones, and then we have hormones of the posterior pituitary, again, just those ADH and oxytocin. If you end up not remembering what kind of hormones are coming from there, pretty much the easiest way to remember it is by remembering that the posterior lobe only has 2, and it's ADH and oxytocin. That way, pretty much everything else coming out of the hypothalamus, you know, has to be coming out of the anterior. So we also have some other hormones that act as neurohormones because they're kind of like neurotransmitters. Well, they are neurotransmitters, but they also act as hormones. That includes dopamine, as we mentioned a little bit earlier, which inhibits prolactin secretion from our anterior lobe of the pituitary. And we have somatostatin, which you saw in your GI chapter. Somatostatin can inhibit the growth hormone secretion from the anterior pituitary. Now, let's talk a little bit about anterior pituitary hormones specifically. So we talked a little bit about thyrotropin-releasing hormone. We said they were secreted by the hypothalamus to the anterior pituitary. They bind to thyrotropic cells using GPCR cell surface receptors. And they use IP3 as a secondary messenger in order to stimulate cells. So they'll secrete thyroid stimulating hormone for T3, T4 in the thyroid gland. Then prolactin, we talked a little bit about that. Prepares mammary glands for breastfeeding, suppresses sex hormones to inhibit menstruation and ovulation during pregnancy. In males, we have excess prolactin can suppress sex hormones, causing infertility in both sexes. Hyperprolactinemia is treated with synthetic dopamine. So we talked about agonists. Again, that would be an agonist, an example of an agonist, that bromocriptine, because it acts just like dopamine and binds just like dopamine for the same purpose, to inhibit the excessive production of prolactin. CRH, we talked about a little bit before. CRH, corticotropic releasing hormone, will stimulate corticotropic cells in the anterior pituitary to secrete ACTH from the anterior pituitary. ACTH will then stimulate cortisol, aldosterone, and androgen secretion in the adrenal cortex of the adrenal gland. Then GHRH, again, GPCR receptors using IP3 and CAMP. Did I already go over this page? I feel like I did. Weird. I did. Is it the exact same page? It is. No, it's not. Okay. I'm sorry. So IP3 and CAMP, a lot of this information repeats itself even in the slides. Growth hormone binds to cytokine cell surface receptors, uses JAK2 and tyrosine kinase, secondary messengers, stimulates cell proliferation and cell growth. GH levels increased in pregnancy and childhood, again, because we are growing a fetus, and then once that child is born, that child also has to grow, so they're under the influence of growth hormone. Excessive amounts can cause gigantism, so that's excessive growth. That'll be a very large child. They may also have an enlarged heart, which is cardiomegaly. Cardiomegaly. Megaly is usually a word that we use to indicate an enlarged organ. So cardiomegaly, enlarged heart. This can actually be treated preventatively if we see that it's a problem in fetal development and childhood development. We can give some synthetic somatostatin, because we said somatostatin inhibited growth hormone. So again, this is a good example of an agonist. So if we have a somatostatin deficiency, we can treat it with synthetic somatostatin as an agonist, because it will act like somatostatin and it will inhibit excessive growth hormone. So on the other side of that, growth hormone deficiency, like we said, could lead to dwarfism, so a lack of growth, which can then be treated with synthetic growth hormone. So this is the agonist for growth hormone, would be just a synthetic version. GnRH, or gonadotropin-releasing hormone, will stimulate gonadotropic cells to secrete follicle-stimulating hormone and luteinizing hormone. did talk a little bit more about this, receptors in the ovaries specifically, if you can see that. Control the development of the follicle and ovum inside during the first 2 weeks of the menstrual cycle, during which time FSH levels increase. We talked a little bit about ovulation by the mid-cycle, because FSH decreases and LH increases. That's a little bit more of what I talked about just a little bit ago, so that information is there for you. Move on. So, again, we talked about FSH and LH in the male, receptors in the testes to control development of the sperm from germ cells. Luteinizing hormone specifically will bind to Leydig cells, so LL, luteinizing, Leydig. Those are together, where it stimulates testosterone secretion, important for the development of testicles in the male fetus, development of external genitalia in the fetus and child, and in the descent of the testes from the abdominal cavity to the scrotum in the fetus. So they do start kind of up here, above your kidneys. So your kidneys move up, your gonads move down. It's a little weird. Testosterone controls secondary sex characteristics. That includes a deepening of the voice, increased muscle tone, and increased libido. Body temperature increases red blood cell production, mood, sleep, appetite, and body metabolism. Testosterone deficiency in the fetus can lead to a failure of testicular descent. And we know that testicles are external because they're sensitive to temperature. So if they never leave the body, they will be too hot, and that will lead to infertility because it will cause a denaturing in those sperm proteins. They will denature and will be useless. So that will cause infertility if the testicles never descend and they stay inside the body. So, talked a little bit about posterior pituitary hormones, including our ADH, antidiuretic hormone, and oxytocin. I already told you about our two hypothalamic nuclei, the supraoptic and paraventricular, which will stimulate their nerve fibers to secrete this into the posterior pituitary. Usually, antidiuretic hormones specifically, which is also called vasopressin, will be stimulated when osmoreceptors in large blood vessels, large arteries, sense that the blood volume is low or that the solute content in the blood is too high. So this could be due to possible dehydration. It could also just be due to dietary things. So those osmoreceptors in the blood and those large arteries are under the control of the vagus nerve, which is cranial nerve number 10, cranial nerve 10, and cranial nerve 9, the glossopharyngeal cranial nerve. Those carry information about our blood osmolarity to the hypothalamic nuclei, our supraoptic and paraventricular, which will then stimulate ADH, our antidiuretic hormone, into the posterior pituitary when we're dehydrated specifically. So ADH is then released into the blood. It will then go to the collecting tubule of the nephron and bind to cell surface vasopressin 2 receptor, vasopressin 2 or V2, on the collecting tubule of the nephron. That will then stimulate the reabsorption of water into the peritubular capillaries surrounding the nephron in order to decrease osmotic pressure by adding water or finding a better balance of solute and water in our blood. So this will increase blood volume if that's the problem, or it will decrease blood pressure if that's the problem. It will also decrease urination because we're absorbing more water and not excreting more water, and it will relieve dehydration that way. Oxytocin, on the other hand, is important in both sexes. In females, it's very important for the development and ability to lactate in the mammary glands. So this will start being secreted in preparation for breastfeeding. It also binds to myometrial cells in the uterus, because we have those 3 layers. One of those layers is the myometrium, which is our smooth muscle layer in the uterus. So oxytocin can bind there in order to control contractions during labor and childbirth. In males, it's just important in the control of ejaculation and vasoconstriction, so no big deal. Adrenal gland. So we can start getting into specifics about our peripheral tissues. First one is the adrenal gland. The These adrenal glands, or suprarenal glands, just in case you end up seeing it spelled, put that way, are located on the upper pole of each kidney. So on the top of each kidney, we have an adrenal gland. Each gland is composed of 90% adrenal cortex and 10% adrenal medulla. So inner adrenal medulla, outer adrenal cortex. This adrenal cortex has 3 layers. That's the zona glomerulosa, which will secrete our aldosterone, or like we said, mineralocorticoid. Our zona fasciculata will secrete cortisol, or glucocorticoid. And our zona reticulata will secrete androgen, androstenedione, and dehydroepiandrosterone, or DHEA. Those are precursors for estrogen and androgen, precursor hormones. And so our adrenal cortex secretion, like we said, was regulated by ACTH from the anterior pituitary, and ACTH was stimulated by our CRH from the hypothalamus. So, and that's just a review on ACTH and the stimulation by CRH down here. Let me get into these specific cortex secretions. Aldosterone binds to nuclear receptors in the collecting tubule portion of the nephrons. Also stimulated by potassium and sodium levels, which will stimulate aldosterone secretion not via the hypothalamus and not via ACTH, but via the renin-angiotensin aldosterone system specifically. So this is another mechanism by which aldosterone is stimulated to be secreted. So this activates when our blood pressure is low, or blood volume is low, or if our sodium or solute levels are low, it will stimulate renin in the kidneys to be secreted. Renin will then convert angiotensin into angiotensin 1, or AG1. And then, through blood circulation, it will eventually make it to the lungs, which have this enzyme, ACE, angiotensin converting enzyme, which will convert AG1 into AG2, angiotensin 2, which will then act as a vasoconstrictor and stimulate the production of aldosterone from corticosterone, which is a precursor, in the zona glomerulosa of the adrenal cortex. Aldosterone is then carried to the blood, then to the nephron, and it will bind to the collecting duct and late distal tubule. This will stimulate peritubular capillaries around the nephron to reabsorb salt and water, absorb bicarbonate, excrete potassium, because again, we're regulated by potassium, and excrete hydrogen ions or acidic waste into the urine. In this way, aldosterone will increase blood pressure and salt blood concentration. And then, let's see, excessive potassium will increase aldosterone, and then aldosterone will then increase potassium secretion into the urine to regulate it and get that hyperkalemia fixed. Cortisol, or glucocorticoid, again, is stress hormone. That's just a review of what we talked about briefly earlier. We can go to androgen, produced in the zona reticulata, binds to nuclear receptors, is male sex hormone present in females. We talked about that. And we also talked about puberty, where it causes axillary and pubic hair growth and also causes acne. So, adrenal medulla. This is our next peripheral tissue. Also part of the adrenal gland. Again, it's that 10% inner tissue. This secretes catecholamines, which are our adrenaline or epinephrine, and noradrenaline or norepinephrine. The adrenal medulla itself is not under the control of the pituitary gland or the hypothalamus, because we looked at anterior pituitary, posterior pituitary hormones, hypothalamic hormones. None of them went to the adrenal medulla. They are, though, controlled by our autonomic nervous system. Pancreas and parathyroid, only part of the pancreas is under the control of the endocrine system. Parathyroid gland, not under the control of the endocrine system, or hypothalamus, sorry. Adrenal medulla is derived from embryonic ectoderm. So these embryonic derivative tissues are something that you are going to need to know. So ectodermal embryonic tissue is neural and gives rise to the central nervous system and peripheral nervous system, as well as some organs and also our skin. Noradrenaline and adrenaline have receptors in the liver, specifically for controlling gluconeogenesis, creation of new glucose, and glycogenesis, the storage of glucose as glycogen. A little bit more specific, norepinephrine produced by chromaffin cells in the adrenal medulla. So chromaffin cells, those are a specific cell type. They're also produced in the central nervous system and peripheral sympathetic nerves. We already know that they are neurotransmitters. We know what they do. We know where to find them. So again, the effects of the norepinephrine depend on the type of receptor it binds to. If you remember, our alpha-1, beta-1, alpha-2, beta-2. Alpha-1 is going to be for vasoconstriction, contraction of smooth muscles, blood pressure increase. Alpha-2 for relaxation, smooth muscle, dilation of sphincters, and vasodilation. Beta-1 for increased heart rate, myocardial contractions, and increased conduction velocity. We talked about propranolol and atenolol in those chapters acting as antagonists or beta blockers against norepinephrine. And then our beta-2 relaxes our bronchi for increased respiration. Albuterol is then an agonist for norepinephrine if norepinephrine is unable to bind. And then epinephrine is also secreted by chromaffin cells in the adrenal medulla, controls blood pressure, glucose formation, and glucose absorption in the liver. If we're talking about the synthesis of these hormones in the adrenal medulla, we're going to use the precursor amino acid tyrosine, which is catalyzed by the enzyme tyrosine hydroxylase to become dihydroxyphenylalanine or DOPA, dihydroxyphenylalanine. An updated version of this will be available to you later, so don't worry too much with the blanks filled in. So this DOPA can then be catalyzed by the enzyme DOPA decarboxylase to become dopamine, which is then catalyzed by dopamine beta hydroxylase to become norepinephrine. And if we want epinephrine, we can do an extra step and catalyze using the enzyme phenylethylamine N-methyltransferase to get that epinephrine. So next peripheral tissue, the thyroid gland. Thyroid gland, located alongside our hyoid bone and the first ring of the trachea, up here. Has two lobes, one left, one right, like a butterfly. Blood supply to the thyroid gland is important because it brings that iodide ion, that precursor for T3, T4. So if we're looking at the inferior lobe of the thyroid gland, it's going to be supplied by the inferior thyroid artery, and then the superior lobe of the thyroid gland is supplied by the superior thyroid artery. So venous drainage then will be that deoxygenated blood. It will be leaving the thyroid from the inferior and superior thyroid veins. And it's not the rest of that blood pathway is there for you. Thyroid nerve supply. Thyroid is controlled by the autonomic nervous system, which includes our parasympathetic and sympathetic. And the thyroid gland itself is derived from embryonic endodermal tissue, which is the innermost layer of that early embryonic growth, those three germ layers. So the internal structure of the thyroid gland, we have a single layer of follicular cells, and these follicular cells will have receptors for TSH, but those receptors will be on the basolateral surface of the cells, so the bottom side of those cells, and those sides will face the bloodstream. Inside, on the other, more internally into the thyroid, we have a lumen or cavity that is filled with colloid fluid. And that colloid fluid contains thyroglobulin proteins, and those thyroglobulin proteins contain tyrosine amino acids. I'm trying to get that to focus. And those tyrosine amino acids are what we use for precursor along with iodide for T3, T4 production. And so when we talk about T3, T4, T4 specifically is going to be thyroxine, and our T3 is triiodothyronine, or triiodothyronine. So most, as we can see here, 99.9% of T4, 99.9% of T3 in the blood are going to be bound to protein carriers or transport molecules. Most commonly, the protein we find carrying them is one called TBG, or thyroxine binding globulin, and this also it helps inhibit the renal excretion of T3, T4 into the urine, so we don't accidentally waste it. So when thyroid stimulating hormone binds to GPCR receptors on the basolateral surface of those follicular cells, it will activate second messengers, adenylate cyclase and CAMP second messenger, which will cause the other side of the cell, the apical surface, the top, to reabsorb thyroglobulin from that lumen and begin synthesis of thyroid hormones T3 and T4. So it takes it out of that cavity. It absorbs it from that cavity filled with colloid fluid. T3, T4 will then bind nuclear receptors on their target cells, meaning they go inside the cell and bind to that nucleus. And these hormones are critical for cell differentiation during development and in maintaining thermoregulation and metabolic homeostasis in the body, which is why when you see hyperthyroidism, patients are usually very thin and insensitive to temperature changes. And in hypothyroidism, they will be typically overweight because of that metabolic homeostasis being shifted in their body. And they will be more susceptible to heat. So synthesis of thyroid hormones. We said before that we use an iodide pump, which is a sodium iodide symport pump to transport iodide into the follicle cells of the thyroid gland. So once TSH has bound, it will activate those cells to uptake thyroglobulin, like we said, and it will also uptake iodide, ion, from the blood, and that will symport along in with sodium. That iodide then will be catalyzed by peroxidase enzyme. It will oxidize itself into iodine, I2. That iodine molecule is now reactive and ready to form inactive forms of T3, T4 by the process of organification. That's that first synthesis process that we talked about, organification. And organification combines those iodine molecules with tyrosine residues from those thyroglobulin proteins. So if we have one molecule of iodine and one molecule of tyrosine, we get monoiodotyrosine, or MIT. If we have two molecules of iodine and one molecule of tyrosine, we get diiodotyrosine. Then these can be combined in different ways to get T3, T4. We have that here, if we can even see it. So while MIT and DIT are attached to the tyrosine residues of thyroglobulin, two coupling reactions occur. So this is the coupling reaction. One MIT and one DIT will give us T3 triiodothyronine. One DIT and another DIT will give us T4 thyroxine. So most of these reactions are stepwise, and they are catalyzed by our peroxidase enzyme. More T4 than T3 is synthesized because T3 is more reactive. T4 will usually go to target tissue and then be converted into T3 at that site and then do its thing. So that's why we see more T4 in the blood. Thyroid hormone physiological functions. They're both active and independent of each other, but they can also collaborate. And that kind of is what I was saying about T4 being more active because it can then become T3. T3 cannot pass through the maternal placenta, but T4 can. So what it does is it passes through and then converts itself to T3 in order to control the development of the central nervous system, bone, and thyroid gland in the fetus. From that maternal blood into that fetal blood supply. More specifically, T3 can synergistically act with growth hormone and somatomedin, not somatostatin, somatomedin hormones for fetal bone formation. And in adults, thyroid hormones are more important in controlling our memory, our learning, our metabolism, like I said, our temperature, oxygen consumption, and a whole bunch of other good stuff. including increasing the sensitivity at our alpha-1 and beta-1 sympathetic receptors. So increasing the sensitivity here will make them more reactive or make us increase the products when these receptors are bound. So we get an increase in ventilation rate, heart rate, contractility of the myocardium, and blood pressure. Now, let's move on to parathyroid glands, which are one of those that were not controlled by the hypothalamus. So these are actually posterior or behind the thyroid. If we have one lobe left and one lobe right, then our parathyroid is 2 lobes left, 2 lobes right, 4 in total, left and right, 2 each. They use cell surface GPCR and CAMP as a secondary messenger. They are sensitive to phosphorus and calcium levels in our blood, and 10 milligrams per deciliter is the normal calcium value. Chief cells specifically. Chief cells are just kind of a general term. We saw chief cells in the GI. Chief cells here, though, in our parathyroid, will secrete parathyroid hormone. Get that to focus. Parathyroid hormone will regulate blood levels of calcium and of phosphorus. So if parathyroid hormone is active in increasing one, the other will decrease. So if we see an increase in calcium, phosphorus will decrease. If phosphorus increases, calcium will decrease. So parathyroid hormone secretion is controlled by a negative feedback loop. So high levels of calcium will decrease parathyroid secretion. Parathyroid binds to bones sometimes, which is why it's important to have a negative feedback mechanism to keep it from doing this. And when it binds to the bones, this process is called bone resorption. Bone resorption, because we are actively removing calcium from the bones, which was previously deposited into the bones to make them strong. So when we remove them, it weakens the bones, makes them more brittle and prone to breaking. But a weird thing about that is, even though we take it from the bones, it's not directly translated into an increased blood level or serum level of calcium, because phosphate will also come with it and be associated. And so what we can do is inhibit phosphate reabsorption in the proximal tubule as well. So phosphate will be excreted in larger amounts through the urine. And once that happens, phosphate levels will decrease, and the phosphate associated with calcium will actually disassociate in order to replenish that lost phosphate from the urine. So that's a way that we can disconnect that calcium from that phosphate and then increase blood serum levels of calcium. And that's what we call phosphaturia. It's right there, phosphaturia. So in order to get that calcium that we take from the bones and put it into the blood, we have to disassociate phosphate from it first. And we can do that by excreting phosphate in the urine. Parathyroid hormone receptors are on the distal tubule of the nephrons. This results in calcium reabsorption. Parathyroid also has receptors in epithelial cells of the small intestine, because that's where the majority, if not all, of our nutrient absorption occurs in the GI. So we can get an increased absorption of calcium there from our chyme. Magnesium is also critical in absorption of calcium. If we have a slight decrease in our blood magnesium levels, it can start an increase in parathyroid hormone. But if we get a massive deficiency in magnesium, this can actually cause an inhibition of parathyroid hormone. And this can be because magnesium is critical to, thousands of different systems in our body. So it could be part of the synthesis mechanism. She doesn't really specify what she needs you to know specifically for that. But just know that severe deficiencies in magnesium will inhibit parathyroid hormone. And this will lead to hypoparathyroidism and hypokalemia, so decreased levels of calcium in the blood, because there's no PTH stimulating reabsorption of calcium. So those distal tubule receptors of the nephron can also convert inactive forms of vitamin D, and that inactive form is 25-hydrocolaglutamate calciferol into an active form, which is 1,25-dihydroxycholecalciferol. And that active form is then absorbed into those paratubular capillaries surrounding those nephron tubules. And this process can generate CAMP as an energy source intracellularly, which is then secreted as waste because we don't need it anymore. And that vitamin D is also vital for calcium absorption. If you are somebody who's not exposed often or regularly to sunlight, you can actually develop a vitamin D deficiency, which will then affect your blood levels of calcium. So getting some sun exposure is important also in the synthesis of vitamin D. Vitamin D then travels to the GI to assist with calcium absorption from those epithelial cells of our small intestine. Deficiency in parathyroid hormone causes a deficiency of vitamin D because, again, parathyroid hormone binds to those receptors in the nephron in order to activate vitamin D to be reabsorbed. So we can get osteoporosis from all this. Deficiency of vitamin D, deficiency of calcium, osteoporosis, because again, if there's no blood level of calcium, it's going to go looking for calcium somewhere else, and that somewhere else is the bone. We don't want that. We want to prevent that as much as possible. So osteoporosis will leave us with brittle, weak, and porous bones. We'll be very susceptible to bone breakage and very poor bone healing. Hyperparathyroidism will also result from this. So, again, some important things about calcium. Important in muscular contractions, seen in very large amounts in the mechanisms of the neuromuscular junction. If you remember that neuromuscular junction from the beginning of the semester. Calcium is used in large amounts in cardiac muscle. Also used in the muscularis mucosa, which is the smooth muscle layer of the GI tract. Calcium regulates action potentials at sinoatrial nodes and atrioventricular nodes, so SA and AV nodes. In hypercalcemia, excess calcium can increase the heart rate by stimulating SA and AV nodes. And we have vitamin D and calcium, important. Pancreas as another peripheral tissue. Pancreas is located between the second part of the duodenum and the spleen. It has exocrine and endocrine function, so obviously the endocrine function is going to be important for us to know. But just a review on that exocrine function, we've got those digestive enzymes, lipases, amylases, and proteases. And then the endocrine portion of the pancreas that specifically secretes endocrine hormones is going to be called our islet of Langerhans. And there are 4 specific cells that you need to know from this portion of the pancreas. That's alpha cells, beta cells, delta cells, and PP cells. Those alpha cells are going to secrete glucagon, beta cells, insulin, delta cells, somatostatin, and gastrin. We talked a little bit about those gastrinomas in the pancreas causing an increase in gastrin that cannot be controlled. That is cancerous. PP cells will produce less than 1% of something called pancreatic polypeptide, which can act like paracrine hormone, so they can act a little bit like somatostatin. And then these cells in the islet of Langerhans, specifically the beta cells and alpha cells, will have gap junctions. So beta cells to beta cells, alpha cells to alpha cells, or alpha cells to beta cells to facilitate rapid cell-to-cell communication. Because if we look, we have glucagon and insulin at the alpha and beta cells. And those usually help to regulate each other in the blood levels of glucose. We can get a little bit more into those specifics. So glucagon, alpha cells. Secretion depends on the blood level of glucose. Low levels will increase glucagon secretion. They use a second messenger camp to stimulate production of glucose from stored glycogen. Glucagon can elevate the blood glucose level by increasing the rate of glycogenolysis, which is the breakdown of glycogen into glucose, and inhibiting glycogenesis, which is the storage of glucose as glycogen in the liver. Glucagon can also increase blood glucose by increasing gluconeogenesis, the formation of new glucose, and this does this along or synergistically with cortisol. There's some molecules that it uses to create new glucose molecules. Lipolysis and proteolysis are very important mechanisms for the breakdown of lipids and the breakdown of proteins in order to generate these molecules necessary for the generation of brand new glucose. Proteolysis and lipolysis. Now, we have somatostatin in the delta cells, which can downregulate or decrease the activity of insulin and glucagon secretion. So it inhibits their secretion indirectly. It can decrease peristalsis as well to prolong the amount of time that chyme spends in the digestive system, and a whole bunch of other stuff that we this pretty much review from your GI chapter. Insulin from your beta cells. Insulin secretion is increased when blood glucose levels are high to facilitate cellular uptake of that glucose from the blood and into the cells. Our normal levels should be around 70 to 110 milligrams per deciliter. 70 to 110. Insulin has receptors in most types of cells in the body because all cells need glucose to function. But more importantly, we see them in receptors for insulin in the liver, muscles, and adipose tissue. Very important here. And target cell uptake of glucose from the blood will facilitate then the production of ATP, energy, in the cells. So hyperglycemia, as we said, when glucose gets too high, increases insulin secretion, while hypoglycemia will increase glucagon secretion. So taking glucose out of the blood, putting glucose in the blood. Hypoglycemia occurs when the blood glucose drops below 50 to 60. When blood glucose is too high, insulin stimulates glycogenesis, which is our glucose to glycogen storage in the liver. Insulin will then inhibit glycogenolysis, which is the breakdown of glycogen into glucose because it's trying to get rid of glucose from the blood, not put it back in. And then insulin will also decrease gluconeogenesis by increasing the levels of fructose 2,6 biphosphate. And this does this by promoting an enzyme activity of phosphofructokinase. So this will decrease also lipolysis, because again, lipolysis and proteolysis give us the molecules needed to create glucose, and we're trying to stop that from happening. So we will decrease lipolysis and increase fat storage instead of fat breakdown. This will decrease ketoacid formation because lipolysis will form acetyl-CoA byproducts, which will then, in general, give us a increased ketoacid or ketone body product in the blood, which we do not want. We don't want acidic blood. So here is a little bit about our insulin secretion mechanism, how we actually get insulin from those beta cells. So glucose is actually, glucose here, is actually going to enter a pancreatic beta cell using a GLUT2 channel. So right here, GLUT2 enters the beta cell with a GLUT2 channel. It's going to close this calcium channel, and it's going to maintain that potassium, and it can also uptake some potassium before closing that channel. This will also stimulate an increase in calcium inside, and that increase in calcium will actually stimulate, I guess, an action potential to the point of it is that it results in the activation of exocytosis, and it will exocytose vesicles full of insulin, which you'll see here. It's not showing up very clearly, but it's a little more clear in your actual paper. It will stimulate exocytosis of insulin into the bloodstream. So that's how we get our beta cells in the pancreas to give us more insulin when there's a large amount of glucose. So all you need to know for that. And then after we get insulin into the blood, that insulin will then turn around and pick up glucose and take it to the target cells to be taken up from the blood. So insulin will then bind to cell surface receptors, and they will activate intracellular GLUT4 molecules. So this GLUT4 right here will be activated. GLUT4 will go to the cell membrane and change its protein conformation in order to become a glucose channel. So it'll open up a pore in the membrane for glucose to come in. So then glucose comes in to the target cell through that GLUT4 channel, and it will be converted into pyruvic acid during glycolysis inside the cell, and then eventually it'll go into the mitochondria for ATP production. And this is what we call cellular aerobic respiration. So, glycogenesis. This just a quick review. Glycogenesis is the creation of glycogen from glucose. We get glucose that goes to the liver. At the liver, it's converted into glucose 6-phosphate, then glucose 1-phosphate, and glycogen. Glycogen synthase enzyme is the enzyme catalyzing this process. Glycogenolysis is the reverse process. That's the storage of glucose as glycogen. No, sorry. That glycogenolysis is when the body needs more glucose. So the liver will break down stored glycogen back into glucose, so we get glycogen, glucose 1-phosphate to glucose 6-phosphate. It is literally the exact same steps, just in the reverse order. And then gluconeogenesis, new, so the genesis of new glucose, is stimulated by glucagon and cortisol, so they're synergistic together. The formation of glucose is from fatty acids, lactic acids, glycerol, amino acids in the liver, which is then carried to the muscle for use. And those lactic acids, glycerols, amino acids, and fatty acids, they are partially coming from that proteolysis and lipolysis. So, pineal gland, that's another thing we can talk about. Located in the posterior region of the third ventricle of the brain, it secretes melatonin to control our sleep. In the absence of light, it stimulates melatonin secretion to induce sleep. As we age, those pineal glands atrophy or calcify, and this can cause sleep loss. So damage in this gland will literally cause us to sleep. That's why old people have dinner at like 4 in the afternoon, they go to sleep, and then they wake up at 3 in the morning. So melatonin synthesis, I don't understand it. I guess I will when I'm 80. So melatonin synthesis, we're looking at a serotonin precursor, which is a neurotransmitter, and that is derived from its precursor, tryptophan. So that's why when we eat turkey, we get sleepy, because tryptophan becomes serotonin, which will then get turned into melatonin because we're eating it at night. So that's a decreased amount of natural light we're being exposed to. Decreased light, increased tryptophan, you're going to get melatonin. Pineal cells will install an acetyl group, an acetyl functional group, in a process we call acetylation, and that's just the installation of this little molecule here, an acetyl. And we will also get an installation of a methyl group in a process called methylation onto the serotonin to convert it into melanin. So this process is dependent on the exposure to light. If we get abnormal light exposure, we will stay up longer because there's an actual process by which our eyes will react to a decreasing amount of light over time, like when the sun is setting. And that's when this increases, almost like a concentration gradient, low to high, the production of melatonin. So when the retina of the eye is exposed to decreasing amounts of sunlight, like when the sun is setting, it stimulates a signal to activate suprachiasmatic nucleus, which is found in the hypothalamus. And that's how it controls our sleep or our circadian rhythm. Those nerve fibers will go down the spinal cord, and they will transmit an activation signal to a superior cervical ganglia in the spine, and that will activate a signal back up the spinal cord and innervate the pineal gland to begin hormone synthesis and secretion of melatonin. So prostaglandins, real quick, bind to cell surface GPCRs of target cells and use second messenger CAMP. In the blood vessels, they are a vasodilator and a vasoconstrictor. In smooth muscle, contraction and relaxation. Blood pressure regulation and inflammation regulation. Lots of stuff, depending on the type of prostaglandin. So we have some examples here: erections, labor, dilation of the uterus, menstruation. It irritates pelvic cavity Prostaglandin E2 specifically can irritate our thermoregulatory set point in the hypothalamus, which causes fever, and typically arises from the precursor arachidonic acid, which is converted with a cyclooxygenase enzyme. We have some common types here. Our PGI2, which is active in antiplatelet aggregation, which keeps us from forming blood clots, unless we need to. PGE2, again, active in fevers, causative agent of fevers, smooth muscle relaxation and vasodilation. PGF2 alpha, is active in smooth muscle contractions and vasoconstriction. And then in combination, PGE2 and PGF2 alpha are both involved in uterine contractions during childbirth. Another one, TXA2, thromboxane A2, is active in platelet aggregation, blood clotting, and vasoconstriction. So we have one that keeps blood clotting from happening and one that causes blood clotting. Prostaglandin precursor arachidonic acid can also be oxidized by a different enzyme, lipooxygenase, to form leukotrienes, which occur inside specialized leukocytes or white blood cells. These are usually active in asthmatic reactions. They induce bronchial inflammation, and all the other things associated with an asthmatic reaction, decreased respiration. So we can say that they act synergistically. Leukotrienes act synergistically in asthmatic inflammatory reactions. And if we want to stop this prostaglandin activity, especially in fever reactions, we can prescribe NSAIDs, which are our non-steroidal anti-inflammatory drugs, like aspirin, ibuprofen, naproxen, and indomethacin, or methicin, which is preferred for babies with heart conditions. But NSAIDs in general, good for prostaglandin inhibition. Now, hormones of the gonads and placenta. Let's just skip all the way down here. Estrogen is secreted by gonads once puberty begins, and then from the placenta during pregnancy. Estrogen in excess can suppress testosterone and cause infertility in males. Estrogen is important for the development of secondary sex characteristics. Let's drop down. I'm going to start moving a little faster. Important for preparing and maintaining the endometrium, the endometrial lining of the uterus, for implanting a fertilized ovum. Secretion is controlled by follicles that develop into the corpus luteum in the ovary during the first trimester of pregnancy. During the second and third trimesters, the control of progesterone secretion is taken over by the placenta, because by that time it's actually developed and grown there. And progesterone deficiency can cause spontaneous abortions or miscarriages. Now, testosterone. Again, we said luteinizing hormone stimulates Leydig cells in the testicles to secrete testosterone. High levels of testosterone can actually inhibit luteinizing hormone secretion via a negative feedback response. And then I think that's actually it for this page, so we're just going to move on because it's already filled out there for you. And now we can actually get to chapter 16, which hopefully we can finish by the time we're done here. Let's start with spermatogenesis. I'm going to stop covering them up, so if you want to jump ahead and fill in blanks before I say it, that's fine. I'll stop covering them. Spermatogenesis, the production of mature sperm, regulated by FSH. During pregnancy, embryo is connected to the yolk sac. That yolk sac produces XY germ cells in the male. Those germ cells will migrate to the gonads, called testes in males. Spermatogenesis, fetal germ cells are converted into spermatogonia A, which stay dormant until puberty. During puberty, spermatogonia will become type B, undergo mitosis or mitotic cell proliferation, mitosis, and then they will grow to become diploid, XY primary spermatocytes, XY primary spermatocytes. Each of those will then undergo their first meiosis, so meiosis 1, a meiotic reduction division, and become a 2 haploid, 1X, 1Y, secondary spermatocytes. They will then undergo meiosis II and become 4 haploid spermatids. 4 haploid spermatids, which eventually will enter spermiogenesis and mature into 4 spermatozoa, sperm. So FSH binds to G protein-coupled receptors. CAMP is a secondary messenger. Sertoli cells will inhibit FSH by secreting inhibin. And then, of course, GnRH will stimulate gonadotropic cells in order to secrete FSH and LH. We already said that earlier. Oogenesis, also controlled by FSH and LH. Development of the mature oocyte. During embryonic development, those XX germ cells will migrate to the primitive gonads of the fetus, which now are the ovaries, during week 4 of pregnancy. Germ cells are then converted into oogonia. They begin mitosis in order to proliferate, but then will stop. So unlike sperm cells, they will stop mitosis, and then they won't ever really start again. And they will then grow after that. They will grow in size into primary oocytes. They will collect an outer layer of connective tissue to become the primordial follicle. They will then begin meiosis I during fetal life, but they will not complete it. They will become arrested in meiosis I. So they pause in meiosis I. That primary oocyte stays dormant inside the follicular cell because the follicular cells are secreting OMI, oocyte maturation inhibitor, until puberty, which at that point in time, that primary oocyte will begin to grow in size again. And that primary, that follicle, that connective tissue will develop into cuboidal epithelial cells and form a primary follicle, then mature into a secondary follicle, into stratified cuboidal epithelia. So they went from a single layer to a stratified layer. During puberty, 1 follicle a month will mature, sometimes 2. That's how we get fraternal twins. They will grow inside the primary, that secondary follicle, and complete meiosis I and become a secondary oocyte. Then it will begin meiosis II, but then again will pause in meiosis II, will not complete meiosis II until fertilization occurs. So they will not ever complete meiosis II unless they are fertilized. If fertilization does not occur, menstruation follows ovulation, and that secondary oocyte is removed from the body. It's excreted out along with that endometrial lining of the uterus. So we get rid of it. Typically 1 a month, sometimes more. So birth control. It kind of creates a synthetic stage of pregnancy, a false pregnancy, which occurs naturally once we reach the ages of 50 to 55, and that's called menopause. Contraceptive pills are typically synthetic estrogen and progesterone, which secretes FSH to inhibit ovulation and to kind of create that false pregnancy state. Follicle-stimulating hormone level increases during phase 1 or follicular phase. FSH then decreases in phase 2, while luteinizing hormone increases. Then luteinizing hormone itself is important for ovulation, which is the rupture of the follicle cell so that the egg can leave. And that egg will migrate out of the follicle and into the ampulla, which is the beginning of the fallopian tube. The ampulla is where it will be fertilized. So, male fertility depends on anatomic structure of the sperm, number of sperm, hormones, contents of the semen. Any change or deficiency in the norm can cause infertility. So if we're talking about the seminal vesicle, and the prostate gland, those both release factors that provide nutrients to the sperm and via the semen. So they help create the seminal fluid. At least 10% of all sperm in ejaculate is unable to fertilize an oocyte due to structural abnormalities or immobility. So 10% of them are going to be abnormal and unable to work. And then our average volume of seminal fluid, and that seminal fluid ejaculate emission is the combination of sperm, seminal fluid and prostate secretions. That's going to be about 3.5 milliliters, and that's going to contain about 100 million sperm. So 90 million of those are working just fine. That's a little scary. So mature sperm structure. Structure is important for fertilization. That head contains the nucleus, which holds 23 paternal chromosomes, 22 of which are somatic, which means body, and 1 is going to be our sex-determining chromosome, which will be X or Y. Once fertilization occurs, we are left with a single-celled organism, which is called a zygote. Oh, sorry, I skipped this one. That head of the membrane, the egg shape of sperm, like this, is because the head here has this pocket in the front that's covered in that acrosome membrane, and that's where all those enzymes are going to be held, because this is the first point of contact against that egg. So that's where all those enzymes need to be, at the very forefront. So that acrosome will contain all of those digestive enzymes to penetrate the egg. Once it penetrates and we have fertilization, we get a zygote, single-celled organism with 46 chromosomes. 44 are somatic and 2 are sex chromosomes, either XX or XY. Now the short arm of chromosome Y will contain the gene that codes for the sex determination of that embryo. Neck of the sperm contains mitochondria to produce ATP. And then in that ejaculate fluid, that seminal fluid, we're going to have fructose, prostaglandins, zinc, and calcium to help provide energy for the sperm and also to soften endometrial tissue to facilitate an easier fertilization. Aspermia is a condition in which the male is unable to produce normal amounts of sperm. And this is usually due to a hormonal issue. So, male reproductive system, signals from the testis and the central nervous system to control reproduction. GnRH from the hypothalamus will stimulate anterior pituitary, which we talked about. Gonadotropic cells will secrete FSH and LH. LH controls testosterone from the Leydig cells. Sertoli cells secrete inhibin. So that's all review. And we do have a little diagram here. But with the amount of time we have, do you want to fill out this little diagram? I will be posting the answers later anyway. So do you want to move on, or do you want to fill out the diagram? Move on? Cool. So that's just some gross anatomy terms. So Sertoli cells will secrete during sex determination in the male embryo anti-Mullerian hormone. Anti-Mullerian hormone. And that the mesoderm layer of the embryo has tubes called those mesonephric ducts here on the inside. And those will contain the gonad in the abdominal cavity before they descend. Parallel to them are paramesonephric ducts. Those are on the outside. Those will help to develop female reproductive organs. Anti-Mullerian hormone is specific for inhibiting paramesonephric ducts from forming female reproductive structures. So anti-Mullerian hormone makes sure that no female genitalia are created, or it's going to try to do that. And it's going to make sure that gonads descend as testicles instead. So a deficiency in anti-Mullerian hormone can lead to intersexuality, resulting in male genitalia and a uterus. Intersexuality can also be caused by aromatase, and we usually see this in overweight patients because that fat tissue can actually contain a larger amount of this enzyme, which will actually catalyze the conversion of testosterone into estrogen, so we can get feminization of the body, breast development, and infertility as well in obese patients, obese males. Erection, emission, and ejaculation. We know ejaculation is controlled by both parasympathetic and sympathetic nervous systems, and that ejaculate fluid is made up of secretions from both the prostate gland and our seminal vesicle. This facilitates accelerated motility, and we already talked about these factors, some of them at least. Fructose, calcium, zinc, prostaglandins, citric acid, and mucus are some of the the new ones we didn't go over earlier, citric acid and mucus, to help facilitate motility and fertilization. So erection is controlled by parasympathetic vasodilation and is innervated by nitric oxide neurotransmitter. Nitric oxide neurotransmitter will stimulate cGMP production, which is a secondary messenger, to increase blood flow in the penile arterioles of the corpora cavernosa tissue of the penis, corpora cavernosa. And if we're talking about the ejaculate pathway, we can start in the testicles where sperm is, and those are contained in the seminiferous tubules. They leave through the epididymis, then to the vas deferens or ductus deferens. Seminal vesicle adds seminal fluid to sperm, which leaves through the ejaculatory duct, and the prostate and ejaculatory empty into the urethra, and then from the urethra we have that final emission. Now, specifically for testis, Sertoli cells line the inner wall of these seminiferous tubules and help regulate spermatogenesis, secretion of FSH levels. These cells do not divide, which is why we get a decrease in male fertility as men age, or a decrease in sperm count, is because these Sertoli cells do not divide, they do not regenerate. So Leydig cells secrete testosterone, and the highest level of testosterone is going to be in the morning, which is why whenever you have your blood tested for hormones, they always do it super early at like 7:30 or 8 in the morning, because they're trying to get that very high reading to see how close you are to normal. So menstrual cycle. We talked about the first phase being the follicular phase. This second phase, the luteal phase, will be when luteinizing hormone increases. During that 13th to 15th day, ovulation will begin 12 to 24 hours after the increase in luteinizing hormone. 12 to 24 hours after. That follicle that the egg bursts out of, before it bursts, it's going to develop an avascular spot, so it's going to take out the blood vessels so that we don't have internal hemorrhaging in the ovary. And that spot with no blood vessels anymore is called the stigma. So that area can rupture open without bleeding, and it will release the secondary oocyte into the ampulla of the ovary. That follicle, once it's been ruptured, will be converted into corpus luteum, which will then start to secrete progesterone and estrogen. And that's going to try to help prepare the endometrium of the uterus for possible fertilization and implantation of a fertilized egg. Secondary oocyte itself, when it leaves the follicle, will be covered in two layers, the zona pellucida and the corona radiata, and it will wait in the ampulla of the fallopian tubes for possible fertilization. So the zona pellucida is the protective layer around the oocyte. That's the first layer around the egg. And it will consist of glycoprotein. It's not really tissue. It's just kind of like a jelly. Corona radiata is the protective layer that surrounds the zona pellucida, and that will consist of smaller follicular cells, so that is kind of a tissue layer. So if no fertilization occurs, menstrual cycle will then proceed to phase 3, the secretory phase. You do not have this phase if fertilization occurs. So if no fertilization occurs, then estrogen levels will plummet, and menstruation occurs to shed that prepared layer of endometrium and the unfertilized secondary oocyte, which is why we have bleeding, because you're literally getting rid of an entire tissue layer from the inside of the uterus. So if we don't have any sort of fertilization, that corpus luteum starts to degrade. Over 10 to 12 days after following ovulation, it will become scar tissue, and that scar tissue is called the corpus albicans. Albicans because alba means white, so we have a white corpse. And the actual phase of menstruation and bleeding should last 5 to 7 days. After menstruation, it starts back up again. Estrogen increases for the regeneration of that endometrial lining and for the development of a brand new layer. So ovulation, days 13 to 15. Again, corpus luteum grows into the corpus luteum of pregnancy if fertilization occurs. So corpus luteum becomes corpus luteum of pregnancy if fertilization occurs, and increases progesterone secretions from what it was already secreting. These secretions are under the control of the human chorionic gonadotropin, HCG, human chorionic gonadotropin, which will stimulate the corpus luteum to maintain specific levels of progesterone throughout the first trimester before the placenta develops. Once that placenta develops, it will take over secretion of progesterone and HCG, and it will maintain the corpus luteum during the first trimester. So, how are we doing on time? We've got 9 minutes. Let's go. Progesterone deficiency in pregnant females can cause spontaneous abortions because, again, that progesterone is what's maintaining that placental development. If we have no placental development, there's no way to give nutrients to the developing embryo. Again, corpus luteum, if no fertilization occurs, will develop into the corpus albicans scar tissue. Female reproductive system, we have an outer layer, the parametrium. The middle layer is the myometrium, that muscle layer, smooth muscle layer, and then an endometrium. That endometrium is full of blood vessels, which is why during menses there is such heavy blood flow during that tissue shedding. And then down here we have the cervix, is the lower opening right around here. So that would be the cervix of the uterus into the vagina. And this would be the cervical canal. So fertilization. Oocyte must penetrate, once the sperm reaches the secondary oocyte, it has to penetrate through the corona radiata surrounding the zona pellucida of the oocyte. Then it can penetrate the zona pellucida. And it does this using those enzymes inside the acrosome. Those enzymes specifically are neuraminidase, and acrosin, which again are stored beneath the acrosome membrane of the head of that sperm. Then they penetrate the zona pellucida after the corona radiata. Once that sperm makes contact with that secondary oocyte after going through both layers, it will kickstart the continuation of meiosis II, and that secondary oocyte will finally complete meiosis II, and then immediately begin the formation of the fusion of that female pronucleus, 23 chromosomes, 22 somatic, 1 sex chromosome, and formation of the male pronucleus, 23 chromosomes, 22 somatic, and 1 sex chromosome. Their nuclear envelopes will degrade, which will allow their chromosomes to unfold and then, jumble together and prepare for mitosis as a single-celled zygote. So we start as a single cell, and then we immediately undergo mitosis for massive cell proliferation. Pregnancy lasts 38 to 40 weeks on the average. Missed periods are a typical sign of pregnancy, but we can do a blood or urine analysis, like those little urine sticks. They're 99.9% effective, to detect the presence of human chorionic gonadotropin, HCG. That will be the chemical marker confirming pregnancy, because that's the chemical marker that's pretty much telling us that a placenta is being developed. Fertilization will take place, and that zygote will undergo mitotic cell division. And once it undergoes proliferation for 5 days, that zygote will implant into the endometrium and has become a blastocyst. That blastocyst will secrete HCG, and it will stimulate progesterone secretion from the corpus luteum, which is still active at this time as the corpus luteum of pregnancy. That progesterone, which will then maintain that endometrial lining during the first trimester to nourish the blastocyst until the placenta has developed, which will be during the second or third trimester again. As cell proliferation continues, after around the first 2 weeks, so that was 5 days, blastocyst. Two weeks, we get 2 tissue layers, one being an embryoblast and one being a trophoblast. So we have embryoblast and trophoblast. That trophoblast layer becomes the placenta. So we lose it completely. Even though it's our cells, we lose it. It becomes the placenta. The embryoblast will then develop into the embryo at weeks 3 to 4, and then becomes a fetus at weeks 8 to 9. During week 3, that embryoblast will develop 3 distinct tissue layers, which give us those organ systems and structures. Ectoderm, the outer layer, central nervous system, peripheral nervous system, epidermis, all this other stuff. Mesoderm, dermis, connective tissue, blood, muscle, bone. and endoderm, GI tract, except for the oral cavity and anus, and some endocrine glands. And again, during the first 8 weeks, it is an embryo. From the 9th week of development and on, it is a fetus. During the first trimester, the corpus luteum controls progesterone secretion, and during the 2nd and 3rd trimester, the placenta controls progesterone secretion. And I think this is our last page of chapter 16, and then we have a couple minutes for immuno. Labor and parturition, oxytocin will cause uterine contractions and the dilation of the cervix. That will allow the fetus to move into the vaginal canal. So the uterus squeezes and the cervix opens to push the baby out. Prostaglandins control labor and delivery. So PGF2 alpha will stimulate smooth muscle contractions and vasoconstriction. PGE2 will stimulate smooth muscle relaxation and vasodilation. Human chorionic gonadotropin binds to cell surface GPCR receptors on follicle cells and uses CAMP as a secondary messenger. It will control follicle cells in the corpus luteum after fertilization to control the secretion of progesterone, like we said, to maintain and prepare the endometrium. Now, HCG regulation of progesterone is important during pregnancy, again, as a decrease in progesterone can lead to spontaneous miscarriage because that placenta is no longer being supported. Now, innate immunity. I believe there were some specific areas of chapter 17 she wants you to focus on. So innate immunity is one of those things. That is a non-specific immune response, meaning that it is equipped to handle any general infection. Anything you throw at it, it can handle. It has defenses inside your body and also on the outside. These defenses are always present and active, and they are the first line of defense against infection. If you are undergoing an infection from bacteria for the very first time, this will be holding down the fort and keeping you alive while your adaptive immune system is creating a response, and that should take about a week. So your innate immune system keeps you alive for a week, long enough for your adaptive immune system to respond. Innate uses 3 major types of phagocytic cells. We have neutrophils, which we know are white blood cells. They will use degradative granules inside the cell for phagocytosis. We have monocytes, which are immature, and they are found in the blood, circulating at all times. When they are mature, they turn into macrophages, and those are usually found in connective tissues. Monocytes can also be recruited to the site of infection and then turn into macrophages once they confirm that an infection is there. We've got all this information here. Chemotaxis is the movement of a immunocell towards the site of infection. They go along a concentration gradient. So the site of infection will start releasing cytokines called chemokines. Cytokines are a class or chemokines are a class of cytokines. Since they release from the site of infection, their concentration will be highest at the site of infection and lower as those signals move away from the site of infection. And those cells that are being recruited to the site of infection know to move up a concentration gradient. So they'll get closer and closer the higher the concentration goes. Neutrophils are the first effector cell to arrive at the site of infection. Not the first cell at all, because there are other cells that are recruited there, but the first immunocell that will be recruited to the site of infection. Monocytes will arrive afterwards and turn into macrophages, again, once they have confirmed infection is there. Adaptive immune, like I said, about a week to develop, and it develops specific to that infection. So if there's a specific species you're being infected with, if there's a specific strain of that species you're being infected with, that adaptive immune response will be created just for that, just like one puzzle piece in a 1,000-piece jigsaw puzzle. It's very specific for that infection. So it will result in the production of a pathogen-specific antibody. And after that infection resolved, it will preserve those antibodies in a memory cell. So if you were ever infected by that again, that memory cell will reactivate, and those stored up antibodies will be reactivated and copied to attack that infection again. Adaptive immune cells include T cells and B cells, which are leukocytes, a specific type called lymphocytes. How are we on time? We are at 10:30, and we had maybe like 3 pages left, so I think for 27 pages we did pretty well. I'm sorry I went so fast, and there were a couple things I kind of had to gloss over a little bit, but I want to thank you guys for coming. So the filled-in version of this will be available to you, don't worry. And again, it is, I mean, I've tried to make it as complete as possible, though it's not at 100%. So please try to supplement with your own notes. If you find that there's something missing, feel free to add it. I tried to make this a Word document and not a PDF, so that if you wanted to add more to it, you could. I want to thank you guys for an excellent semester. Once more, since we are out of time, I'm going to go ahead and let you guys know that tomorrow you have another session with your SI leader, Vasily, and that will be here in the Student Union as well. The rest of your study materials, including a clinical guide and a video playlist guide, are on the normal website here. And if you need an escort back to your car, there are 2 numbers here for you to call. They will send a golf cart or a security officer to escort you back to your car. And I don't know if there are any evaluation forms still floating around, but if you did sign one or fill one out, just bring them to the front table, or I'll pick them up when you leave. But thank you guys so much for coming. Good luck for studying.